this video we are going to see about the experiment which is conducted by Alfred Hershey and Martha Chase to prove the DNA as a genetic material. During the late 20th century, even though the researchers have identified the factor which is responsible for the inheritance of characters comes from within the cells, but they have failed to prove the DNA as a hereditary material. So the question for the genetic material remains unanswered. And it was confirmed by the Griffith experiment, but it is not widely accepted, and further the hereditary material is isolated by the experiment conducted by Avery and McCarthy, but this is also not well published. And the final experiment is an experiment conducted by Alfred Hershey and Martha Chase, and this is the only experiment which has confirmed the DNA as a genetic material. So the experiment begins by cultivating viruses in two different medium. So we have viruses, that is the bacteriophages, in two different medium. Let us consider the medium as A and B. In the medium A, the protein is made radioactive. How is it possible? Means by making the radioactive, by adding the radioactive sulfur here in this medium, we are adding radioactive sulfur. In medium A, we are adding radioactive sulfur, and in medium B, we are adding radioactive phosphorus. So here sulfur is made radioactive and it is added in the medium, and here the phosphorus is made radioactive and it is added in the medium. After adding this sulfur and phosphorus, here we can find that since radioactive sulfur is added, the protein is made radioactive because the protein is a sulfur based component, and here by adding the radioactive phosphorus, the DNA is made radioactive because DNA is of course the phosphorus based component, and so here the DNA is made radioactive. So after radio labeling, this bacteriophage is allowed to infect a E. coli cell. So this protein radio labeled bacteriophage is allowed to infect a E. coli, and this DNA labeled phage is also allowed to infect a E. coli. So both phage infects the E. coli, and this media is now centrifuged, and so this is called as blunder experiment. This experiment is otherwise called as blunder experiment. So after infection, this is subjected to centrifugation. So after infection, we are subjecting the medium to centrifugation. So after centrifugation, here in the protein radio labeled phage, here we can find the supernatant is radioactive. Radioactivity can be measured in the supernatant. And here in this case the DNA is made radioactive, and here we can find the radioactivity in the pellet. After centrifugation, as you all know, there will be separation as pellet and supernatant. Here radioactivity is measured in the supernatant, and in this case the radioactivity is measured in the pellet. So why is it so? Because here, since the protein is radio labeled, and this is not the hereditary material as we all know now, and it is not the hereditary material, and so it is not transferred from the bacteriophage to the E. coli, and so no radioactivity enters the cells, and no radioactivity can be detected in the cells which is settled down in the pellet. And here, since the DNA is a hereditary material, the genetic material is transferred from the bacteriophage to the E. coli cells, and here radioactivity can be seen in the cells, and so the radioactivity here it can be detected in the pellet which is settled down in the media. So here radioactivity can be seen in the pellet. So what this experiment confirms is, so the protein is not the genetic material because it is not inherited from the bacteriophage to the E. coli. Also here the DNA is a genetic material, and so it has been inherited from the bacteriophage to the E. coli. So the take home message from this video is, here from this experiment it was confirmed that the DNA is a only hereditary material which is transferred from one generation to another generation, and this experiment has been published also. Thank you.